It's okay. Who is doing the recording?
স্যার আমি দানিয়েল, মোহাম্মদ দানিয়েল স্যার।
Anil, okay. Okay. Today we are going to discuss the next topic, that is PCR, polymerase chain reaction. And this is a very useful protocol. You cannot find any biological lab which is not using polymerase chain reaction. It is useful in almost every different types of biological lab because of its simplicity and what should I say? Simplicity and accuracy, and also the time it required is not that much. You can get a single result within 3 hours. Usually 2 to 3 hours is good enough to get an idea about your result. Now, why people are doing PCR? There are many different reasons of doing the PCR. The most common reason is to find out a specific genetic sequence or DNA sequence in your sample. For example, you have many different types of sample and you have a question in your mind that whether this X gene or any specific gene is present among your 20 samples. And you have to design a PCR so that you can identify whether those 20 samples are present in your samples or whether those 20 samples contain that gene. For that, you have to have a knowledge about the sequence of the genetic sequence of the gene, and you have to design a specific pair of primer, usually the forward and reverse primer, and that forward and reverse primer with some other reagents or some other components, you can design the polymerase chain reaction. And at the end, it will amplify the quantity of the gene product in vitro. In vitro means all these things should be done inside the laboratory. And it is also just, you can name it as just like a photocopy of anything you are looking for. And the formula for the amplification is kind of like, this is the formula. If your final product is, your final product would be something like initial product into 2 into 10 to the power cycle number. Like your, how many cycles you are running, that number would be at the top of 2. So you can get an idea about what would be the product. For example, if you have a single template, there is a calculation here which you could find, like, where is it? This one. From a single template, like this is just a single template, after having, like this is the first cycle, second cycle, third cycle is going on, and with a single template, just a single DNA molecule, you can get this amount, this huge amount. I think it is about 260, 26.8, about 27 million copy of your amplified product from a single template. Usually we are adding not a single DNA fragment as template. There should have like a thousands or like 10,000s or millions of template or millions of fragments as template. So from a single DNA fragment, you can get like 26.8 or about 27 million times amplification after 30 seconds. So you can see this amplification are increasing exponentially. Each and every cycle you are getting exponential increment of your previous product. So that's why it is very popular. Like if you have a very tiny amount of sample or DNA as the template, you can even amplify from that. So that's why it's very popular in most of the biological laboratory. And in our case, DNA from the crime scene is often limited to both quantity and quality. In our case, DNA from the crime scene is often limited in both quality and quantity. Means like we don't have may have like a high quantity of DNA, and also we may have like high quality of DNA. For that, we can use PCR as a very good tool compared to the other protocol that we have, that is that is like RFL. So that's why people are preferring PCR over RFLP for our purpose, and our purpose is to doing the DNA fingerprinting. So to start with the PCR protocol, so these are the just general idea or the thing that you have already known, that in the PCR there are three specific steps. For example, the denaturation. This is your complete DNA, or just take it as your template as a double-stranded DNA. It is denaturation. Then the DNA gets denatured, and then you have to add the oligonucleotide primer. This you can name it as forward primer and it as reverse primer, and then it will be going to be amplified in each and every cycle. So this DNA synthesis at 74 degrees. Usually the denaturation temperature is high, about 94. Then the annealing temperature for the primer is less than that, usually by between 50 to 60. And the DNA elongation temperature is about 72 to 74, between this, because the DNA polymerase that is used here is the TET DNA polymerase, which is very which is a heat stable DNA polymerase, and that's why it can act very good, or it is very active at high temperature. So just take a denaturation step where you add your template 94 degree for 2 to 5 minutes. It is for the first cycle, the time is about a bit more, like 5 minutes. If you take it as 5 minutes, for 5 minutes denaturation is important. That can completely denature your DNA. Completely denature your DNA. And if you are doing the hot start PCR, for that also a long time denaturation is required. After that, each and every cycle, you don't need the denaturation step to be this long. Just 30 seconds is good enough. But for the first step, it is out of the cycle. As first step, you have to denature the samples for a bit long time, about 5 minutes. This is for the complete denaturation of your sample. Then in case of the next step, as in the cycle, the default step is the annealing. Whatever primer you are using, that will anneal to your DNA structure or to your denatured DNA, and this default temperature is usually 55 degrees. So it could be less than 55, it could be more than 55, depending on your primer length or depending on your melting temperature of your primer. If your primer's melting temperature, just for say, is like 50, or no, I should say 60, then your annealing temperature should be at least 1 to 2 degrees less than that. Just for example, the after the primer to be now, if you get a primer with a melting temperature 60, so your annealing temperature should be 58 or 59. Must be less than 60. It could be 50, it could be 52, no problem. But the criteria is it could not be 60, because at 60 degrees, that primer will not remain attached, as it has a melting temperature of 60. We have to reduce the temperature by knowing the melting temperature. If it is 60, your melting temperature must be less than that. If you have a melting temperature of a primer is like 55, you have to decide the melting or annealing temperature for that primer is less than 55. Could be 54, 50, 51, whatever is good, but not 55. And you can go, or you may have to reduce the annealing temperature even for like 40 to 45 degrees, depending on your primer. In that case, you need to, like a 40 to 45 is also may be required, but in that case, you are going to encounter, or you are going to have a problem of non-specific annealing. Non-specific annealing means as the temperature is very low, your primer can non-specifically bind with any of the DNA sequence. I should not say any, partially similar or partially complementary sequence. Just take the primer length of like 30 base pair. At that low temperature, your primer non-specifically can find 10, 15, or 20 base as exact complementary and can bind there. And that can induce non-specific annealing and ultimately non-specific amplification. So the default or the good setting of temperature is about 55. At this temperature, usually only the complementary DNA or primer bind to this complementary DNA only, not to any other specific or not to any other non-specific sequence. That's why annealing temperature is very important. Next, after having the denaturation done, then you have done with the annealing. The next is the extension. Generally, it is 72 degrees centigrade, is between 72 74 because this is the operating temperature or optimum temperature for the thermostable DNA polymerase people are using. People are using the DNA polymerase that we are using usually is stacked DNA polymerase, and it has a optimum temperature of 72. Now my question to you that as it has optimum temperature of 72, do you expect this DNA polymerase to function at room temperature? Just take the room temperature as like 30 degree or 32 degree. Is it possible for this DNA polymerase to do the polymerization at room temperature? What do you think? Sir, no, it is not correct. It will also do the polymerization at low temperature, like the room temperature, but the efficiency will be less. As 72 is the optimum, like you can take it like at 72 degree, it is 100% efficient. But at less temperature, it will also do the polymerization, but with less efficiency. Just take it as like 60% efficient, not 100%. Everybody understand this, that this polymerase has an optimum efficient temperature is 72, but in other temperature less than 72, it will also do the polymerization, but with less efficiency. Any question about this? Okay. If there is no question, so these are the three basic steps, and you have to continue these steps one by one, and depends on the number of the copies of the template DNA and the desired amount of amplification, you can choose the number of cycle. It is between 25 to 35 cycles. 35, more than 35 is not essential. If even after 35 cycles, you are not getting any amplification, you can assume that the PCR that you are trying is not going to happen. So 35 cycle is more than efficient or more than sufficient, and you can choose in between 25 to 35 cycles to get your product. To get your product, 25 to 35 cycle is very good enough. After this 25 to 35 cycles, you can get your desired product very efficiently. So one by one cycle, you are going to get the amplification of your product. Each and every cycle, the product size is increasing exponentially. Any question up to here?
স্যার একটা কোশ্চেন স্যার, তাইলে কি আমরা হলো মেল্টিং টেম্পারেচারটা আগে বের করার পরে দেন আমরা অ্যানেলিং টেম্পারেচারটা ঠিক করি?
Exactly. This is very good question that we need to know the melting temperature. That is for sure. And whenever you are ordering the primer to any company, like we are not preparing the primer in our laboratory, or most of the laboratory do not prepare the primer in theirs. The library or the laboratory are usually ordering the primers towards the other companies. They will provide you the primer, and in that primer tube, it will be clearly mentioned the melting temperature of that specific primer. সবসময় এটা লেখা থাকে, like you order like X gene forward, X gene reverse, and you are going to get two vial or two tube for that. In one tube you have the forward primer, in other tube you have the reverse primer. In forward primer it will be written about the melting temperature of that. It could be like 60 degrees, and the reverse primer may have a melting temperature not 60, could be like 55, 54, 58, whatever, and could be 60 also. So both the primer should have two or same melting temperature. Now, to question to you, get an example. You have a primer set. One has a melting temperature of 60, another has a melting temperature of 58. What could be the annealing temperature of this PCR? এই পিসিআরটা হইতে গেলে annealing temperature কত দিতে হবে?
স্যার 58 এর থেকে কম।
Exactly, very good understanding. You should choose the lower temperature. You should choose a temperature which is lower than the lowest. Like in this case, it is one is 60, another is 58. You should choose lower than the lowest. The lowest is 58 here, so it should be, for this PCR to happen, the annealing temperature should be at highest 57. 56, 55, 54, everything is good, but you cannot do the PCR with 58 degrees centigrade because one of the primer has a melting temperature of 58. Okay?
Yes, sir.
Okay. Any other question? No. Okay, I will take a 5-minute break. Sorry, I was in another call. শুনতে পাচ্ছ?
জি স্যার।
Okay. এখন দেখো, what do you need for the PCR? PCR এর জন্য what are the components that people need to do the PCR? The first thing is template DNA. Whatever the thing that you need first as template DNA. Template DNA means that from where you want to know whether your gene is present or not. That is called the template DNA or sample DNA. It is because this DNA should contain the target sequence which you want to amplify. Template DNA is the first thing that you require. Second is primers. Primers are the short oligonucleotides that define the ends of the target sequence. Ends of the target sequence means whatever the sequence you want to amplify, it has a forward and it has a like upstream end and downstream end. According to that end, you should have primer. And that primer will amplify in between sequences. Then you need a thermostable DNA polymerase. As all these reactions are going on like 72 degree, 94 degree, the lowest temperature is like 50 degree for annealing. So it's very high temperature. So you should have a thermostable DNA polymerase. And we find this thermostable DNA polymerase from bacteria. The very well-known bacteria from where we get this thermostable DNA polymerase is Thermophilus aquaticus, from where the polymerase that we have is called TAC DNA polymerase. Next thing you need is buffer. Buffer is essential for any of the enzymatic reactions. Enzymatic reactions, is this an enzymatic reaction? Yes. As a DNA polymerase or an enzyme is involved here, it is an enzymatic reaction. Any of the PCR reaction are usually enzymatic reaction, and buffer is essential, as we know that for any different types of enzymatic reaction, buffer is necessary. In this case, we also need buffer. Buffer will resist the change in pH. I think all of you should know this, what buffer is doing. Buffer usually resists the change in pH. Drastical change in pH may make your enzyme, in your case it is DNA polymerase, inactive. So buffer will reduce or buffer will resist the pH change so that your enzyme can work properly. Next thing is DNTPs. DNTPs means deoxynucleotide triphosphate. How many DNTPs we have? Someone answer.
Sir, 4 sir.
4. And why we need to add DNTPs here?
স্যার ওই যে ডিএনএ যখন এক্সটেনশন করবে তখন স্যার একটা করে নিউক্লিওটাইড এসে অ্যাড হবে আর এক্সটেনশন করতে থাকবে।
Exactly. So as you are producing or you are amplifying new DNA, so you need nucleotide triphosphate DNTPs to make that new DNA. That's why DNTPs are essential. Next is magnesium. Magnesium plus 2. It is the people are using magnesium chloride as a source of magnesium. Why magnesium is necessary? Because we know that many of the enzymes require this type of metal ions for their activity. They are called cofactor. And in this case, the thermostable DNA polymerase or most of the DNA polymerase that we have are using magnesium as the cofactor. If you do not have magnesium, do you expect DNA polymerase can do its function? Answer.
স্যার করবে হয়তো এফিশিয়েন্টলি করবে না স্যার।
আগেরটার সাথে মিলে তুমি অ্যানসার করছো, that's very good. It will not function at all. Without magnesium, if there is no magnesium in the solution, the DNA polymerase should not function. কোনো মতেই কাজ করে না। ঠিক আছে? So magnesium is very essential. So in any of these type of enzymes which require any of these type of cofactors, that could be magnesium, that could be calcium, that could be any other things, if you can remove that ions, you can make those enzymes inactive. As we know that Chilex protocol for DNA extraction, we remove all the divalent cations by Chilex. So this type of magnesium, calcium, zinc, whatever is present there will be chelated out, and nuclease, which will degrade the DNA, which also require magnesium, will not function and your DNA remain safe. So that's why these type of ions are very important. If you do not add it, you are not going to expect any polymerization. So buffer is essential for pH maintenance. DNTPs are required for the newly synthesized DNA, and magnesium is required for the activity of the DNA polymerase. Now, if you add NTPs, not DNTPs, what is the problem? Do you know between the difference? Do you know the difference between the DNTPs and NTPs? সবাই কি জানো এটা?
না স্যার।
NTP হলো নিউক্লিওটাইড ট্রাইফসফেট আর DNTP হলো deoxynucleotide triphosphate. If you go to the structure of the nucleotide, DNTP, the ribose sugar, do not contain hydroxyl group at 2' position. That's why, as hydroxyl group is not there, it contains only hydrogen. That's why it is called deoxy, oxygen removed. And in case of DNA, all the nucleotides are usually deoxynucleotide. So if you want to synthesize DNA strands, you have to add DNTPs. If you want to synthesize RNA strand, you have to use NTP. NTP means nucleotide triphosphate, where the ribose sugar is usual or normal. That means the 2' position contain hydroxyl group. But in case of DNA, the 2' position do not contain the hydroxyl group. That's why these are called deoxynucleotide triphosphate. Now everybody get it?
জি স্যার।
ডিঅক্সি ডিএনএ আমাদের যে নাম করা হচ্ছে ডিএনএ, যে ডিএনএ মানে হলো ডিঅক্সি নিউক্লিক অ্যাসিড,
রাইবোনিউক্লিক অ্যাসিড স্যার, ডিঅক্সিরাইবোনিউক্লিক
deoxyribonucleic acid. Yes, exactly. Deoxyribonucleic acid. Deoxy terms is for those ribose which do not contain 2' hydroxyl group but contain hydrogen. And in case of RNA, it is ribonucleic acid. Ribose is usual there. Ribose contain all the hydroxyl group as normal. Okay. So if you add NTPs, DNA polymerase will not recognize NTPs as its substrate and it will not do the polymerization. That is the problem. Do everybody get it? যে তুমি যদি, if you in this case do not add DNTP but add NTP, there are NTPs also available. You are not going to get polymerization because DNA polymerase recognize DNTPs as its substrate, not the NTP. Okay. RNA polymerase recognize in case of RNA synthesis, you have to add NTP but not DNTP. Okay. Okay, next is you need a thermal cycler. Thermal cycler means it is a cycling device which will change the temperature within a very short time. Like this cycler or this PCR machine will increase the temperature at 94, then it has to decrease the temperature at 55 for the next cycle or next step, then it has to increase the temperature again to 72, then again go for the next cycle, increase to 94. So the thermal cycler in cycle change the temperature of the machine, and that's why it is called thermal cycler, or we know it as polymerase chain reaction or PCR machine. Without a thermal cycler, is it possible to do DNA PCR? What do you think?
স্যার পিসিআর মেশিন ছাড়া তো স্যার পিসিআর পসিবল না।
Now I am giving you the status that when at 1982 PCR techniques were described or reported, at that time 1982 or just before that 1982 it get the Nobel Prize for having this PCR done. So before or at that time people don't have the thermal cycler, but they report how to do the PCR, and it get attention and get the Nobel Prize because people know it is of immense importance in biology. Then how they did polymerase chain reaction or PCR at that time without thermal cycler? Any idea? This one. This is the machine that was used during that time. It's just a simple water bath. For example, in this water bath, you have 94-degree temperature water. Here you have 55-degree temperature water, and here you have 70-degree temperature water. And you or students like you just change the tubes from here to here to here and again here, then here. This is the cycle going on at that time. By having this just simple water bath of 3 different temperatures that you need, you can even devise a PCR machine in normal lab or in the lab where you don't have the thermal cycle. This machine was the first PCR machine people used. Just 3 different temperature water baths. Now you can understand that without thermal cycler, it is possible. You have to do or you have to change the tubes manually one by one. বুঝতে পারছো রে ভাই এটা কি ছিল তখনকার সময়?
জি স্যার।
So it is possible. Technically, if you can change the temperature by any means, you can do PCR reaction 94, 50, 72. Then again 94, 50, 72. Go this cycle one by one for 25 times. PCR done. Okay. Now, these are the things that we need: template DNA, primers, thermostable DNA polymerase, buffer, DNTPs, magnesium, and then a thermal cycler. Okay, now choosing the primers. How you can choose the primers? Primers should have some specific characteristics so that it can act as a very good device. And where is at the wait, let's see this board. এটা সামনে যেতে হবে। No, not this one. Sorry. This one. Okay, when you design a primer, you have to look for these are the general guidelines for PCR primer designing. For example, the length. Primer length should be in between 18 to 30 bases. 18 থেকে 30 base. Why not less than this? Why not more than this? Any idea? Okay. Why not more than this? It is important to understand that primer designing and preparing the primer or producing the primer is costly. So you don't want to waste your money by designing a primer for like 40 base pair or 50 base pair long. You can argue that more the length, more the specificity. Yeah, that is true. But by having the bioinformatics analysis, it was found that with 30 base pair, you can achieve almost 99.99% accuracy. So it's okay to go with 30 base pairs. If you increase like the same PCR primer, not 30 but 50 base pair, you're going to get the same result. So why you waste 20 bases cost there? you don't need to waste that much. So 30 base is very good enough. With this, you can get 99% accuracy. And 18 base pair should be like it is 18 or 17 in different books. You can find it like 15 also. So no less than like 15 or 18 is because if you reduce it, like you can think of like a primer of like 10 base pair, and you can say that 10 base pair is give you accuracy, no non-specific binding. But it was found that that 18 to 15 is better if you reduce the more smaller the length of the primer, the more increase the chance of non-specific annealing and more chance of getting non-specific amplification. So you should not reduce it like 15 or 18, and you don't need to increase it more than 30. That's why these are the optimum values in terms of the length. Now come to the melting temperature. Melting temperature, this thing is very important. It should be less than 72 because the temperature you are using for polymerization is 72. So it should be less than 72. And it should not be less than like 55 or 50. If you reduce the temperature, the chance of having non-specific annealing get increased as we see before. So it is between 50 to 72 or 55 to 72 is better. This is high temperature. At this temperature, only complementary DNA should bind very firmly. Non-specific binding should be avoided by this temperature. Next thing is percentage of GC content. GC content or GC contented percentage, you have to have a like at least 40 to 60%. So 40 to 60%, the idea is here that GC binding, G or C bind to each other with 3 hydrogen bonds compared to the AT. How many hydrogen bonds are there between A and T? কেউ কি জানো?
দুইটা বন্ড থাকে আর জিসিতে তিনটা বন্ড থাকে।
Exactly. GC-তে 3টা আর AT-তে. In case of AT hydrogen bonding, it is only 2. In case of GC bonding, it require or it has 3 hydrogen bonding. So you can think of if you have more GC, you have more power between the annealing or between the primer and template binding. More GC, more hydrogen bonding, more strong binding. That's why you should have a very good amount of GC content. It is between 40 to 60%. If it is like 10% or 20% GC, you can expect the low strength in binding between the primer and the template. So it should be 40 to 60, more GC, more hydrogen bond, more strong binding. Next is no self-complementaries. Like if you have a primer which contain, for example, this one, at the 5' end, this is the 5' end, this is the 3' end. At the 5' end, if you have 3A or 4A or 5A, whatever sequence, which is exactly complementary to some of the 3' end sequence, you can expect this type of hairpin structure. You see this A will anneal with T, this A with anneal with T, this A with anneal with T. So it's not necessary that it should be A or TTT or GGG or CCC. It could be anything, but it should not have this type of sequence at the 5' side and the 3' side, so that they can make this type of hairpin structure. If it make this type of hairpin structure, this primer is useless. So you cannot use this primer for the amplification purpose. So if you have all your primer like this, you may end up with no PCR amplification or very low PCR amplification because they make the hairpin structure, they are not linear, they cannot bind to the template structure. That's why. ঠিক একইভাবে no complementary to other primer. Like you have one primer and another primer, and in between these two primer, forward and reverse, they have highly complementarity. Then they can anneal with each other. And then as both the primer annihilate each other, they are not useful for the amplification. That's why you also have to keep in mind that these two things, the upstream, downstream, there should have no complementary sequence, and in between the two primers, there should have no complementary sequence. Any question here?
মানে স্যার দুইটা যে প্রাইমার আমরা এখানে অ্যাড করতে চাচ্ছি, এই দুইটা প্রাইমারের সিকোয়েন্স যাতে কমপ্লিমেন্টারি না হয়, আদারওয়াইজ দে উইল
Exactly.
গেট বাইন্ড টু ইচ আদার।
Exactly. মনে করো যে তুমি if you have your forward primer like AT, GC, CC, whatever the sequence, and your reverse primer has exactly complementary sequence there, like 10 base pair of forward primer exactly complementary to the 10 base pair of reverse primer, they will annihilate. And those two primers, ultimately almost all the primers are useless. বুঝতে পারছো?
জি স্যার, বুঝতে পারছি।
Okay, next thing is distance between the two primers on target sequence. Like, what is the size of the PCR product? What is the size? You should not have a PCR product like 10 kilobase, 20 kilobase. The polymerization of this type of DNA polymerase are not that stable. They will not polymerize so long sequences. So you have to have a desired length less than 2000. Like usually most of the cases, what we have 300 to 700 or 300 to 800 base pair is very good enough. Because if you want to know whether your gene is present, just for example, your gene size is, get an example like 20 kilobase. Now you don't need to amplify all the 20 kilobase to know that whether that gene is present in your sample or not. You just have to find a very specific sequence among those 20 kilobase, which is very, very unique for this gene, which is not present in any other sequence in any other human, in any other organism in the world. Just that could be like a 200, 500, 600 base pair. You just have to use that base pair or that length to get an idea or to get a confirmation about whether the gene is present. You don't need to amplify like 20 kilobase gene, you have to amplify 20 kilobase. No, not necessary. 500, 600 unique sequence is good enough. Next is unique oligonucleotide. That's the same thing. It must be unique. Any of these PCR should be very, very unique. You, like if it is not unique, like that sequence or the primer that you're using can annihilate many other different types of sequence, you are not going to get specific amplification. Very, very non-specific amplification you can get. And next thing is TM difference or melting temperature difference between forward and reverse primer. It should not be like get an example, forward primer has a melting temperature 50 and reverse primer has a melting temperature 60. Temperature difference between these two is about 10 degrees. Not good. You have to reduce the temperature difference less than 5 so that you can design the annealing temperature or what or define the annealing temperature very specifically. Okay, like at like 50 and 60, huge difference. You have to design or get the annealing temperature at least 49 according to this. At 49, the 60-degree annealing temperature containing primer may be non-specific bind to other. So this, you have to have the temperature difference less than 5. So one could be like 50, the other could be like 53, 54. Should not be 55. And no long runs with the same base. Like as you see here, this one, AAA. Okay, no long runs with the same base, like AAAA, like you have more AAA. এটাকে কিভাবে লিখবো? ট্যাপ দিয়ে দিই। এরকম যেন long stretch of same nucleotide should not be there. Long stretch of same nucleotide. Same nucleotide, just get an example, A, মনে করো 6টা 7টা A আছে. It should be less than 4. If it is there, you cannot avoid any of these repeat sequences, A, or C. You should avoid those sequences in designing primer. But if you have to take those sequences, please keep in mind that it should be less than 4. একসাথে 4টা A, at once 4A, at once 4T, 4C, 4G is not good. These are the character, or these are the things you have to consider during PCR primer designing. Any question about this? Okay, if no question, then we get an understanding that the PCR primer, to design a PCR primer, it is important to consider those things. Now, see this one, primer dimer. It's a very common problem during what? During PCR. Like when you start to doing your PCR and you're preparing all your samples. Samples means your bacterial samples or could be any other samples. You have the PCR mix, you have the buffer, you have the DNTP, everything there, and you have to prepare like 20 reactions. So each and every reactions to prepare takes some time. So to prepare 20 reactions take a time like a 30 minutes. So first reaction may take just 1 minute, second reaction another minute. So in this way, you may need like 30 minutes to prepare 20 reactions. So when you have to prepare, when you have done with this, the first reaction, second reaction, or those tubes already set on the bench for 20 minutes or 30 minutes or 25 minutes or 15 minutes. And that is very good enough time for the non-specific amplification or for the primer dimer formation. Because as I already discussed, that DNA polymerase that you are using is also active at room temperature. So like take a primer of yours is like this one is like forward primer and this one is the reverse primer. And they must have A and T or G and C. They must have. And those A and T of the both primer can anneal, can hybridize to each other. And this is a substrate for the DNA polymerase. And as you know, at the room temperature, DNA polymerase can function very well. It can then polymerize this part and this part and make a primer dimer, even at room temperature. As we know that the DNA polymerase is active to some extent, not 100% efficiently, but it can do the polymerization. And as we know that the primer must anneal even with a single nucleotide. It is possible. You cannot find a primer that do not contain any A, that do not contain any T. So both primer must contain A or T, must contain G or C. So at least with single base, they can hybridize together. And that hybridization, like this, it can induce the DNA polymerase to bind here and do the polymerization and make this primer dimer. You see, these are the primer dimer. And as these reactions produce huge amount of primer dimer, these primer dimer are now useless. You cannot use this for amplification. And that is a problem. And that is a problem for each and every different types of PCR. Primer dimer is must. Sab সময় হয়. Primer dimer is must. Whenever you add the DNA polymerase, DNTP primers there, they will start forming the DNA primer dimer. So at the end, you are losing many of your primer, like 50%, 60%, 70% primer make primer dimer, and you are losing 60, 70% of your primer. So you don't have those. You have maybe like 20%, 30% left, and that can amplify your DNA with very low efficiency. So that is a problem for common different types of PCR. What can you do? Any idea to avoid this primer dimer formation? Primer dimer formation টা বুঝছো কি জিনিস? আগে একটু বুঝি।
জি স্যার। স্যার প্রাইমার ডাইমার ফরমেশনটা হচ্ছে স্যার যে পিসিআর করার পরে স্যার হচ্ছে যে সময়টুকু
করার পরে না, পিসিআর করার শুরুর সময়, when you start doing the PCR reaction preparation, that time you are adding primer, you are adding what? DNA polymerase, it will start the primer dimer formation at room temperature. Because we know that the polymerase is active at room temperature. Any this type of sequence, any this type of what? Structure where they have annealed with just a single nucleotide, 2 or 3 nucleotide. They have found complementary and they bind in case of the primers. This is forward primer, this is reverse primer. They can bind at A and T, at G and C, or whatever complementation they have, they can bind. And here, DNA polymerase bind and do the polymerization and make this primer as dimer. And this primer dimer are usually long, because if a primer is like 30 base pair, primer dimer is usually 60 base pair or more than 50 base pair. So you have this 50 base pair as useless thing for your PCR. এটুকু বুঝলাম কিনা আগে বলো।
জি স্যার।
Okay, if you do understand this, how to avoid this type of polymerization during your PCR preparation? Any idea? Anyone can answer? কি করা যেতে পারে? You can do a very simple thing to avoid this. You just have to know what you have to do. Stop the polymerization or stop the DNA polymerase to function. When you put your tube on the PCR machine, then it will start. Otherwise, it will not work. So you don't have to worry about the primer dimer, because primer dimer are usually forming during the PCR preparation. PCR preparation may take 20 minutes, 30 minutes, good enough for forming the primer dimer. But at that time, you have to stop the polymerization. You have to stop the polymerization by any means. যে কোনো ভাবেই হোক বন্ধ করতে হবে. What we are doing, আমরা কি করি to stop the primer dimer in our case? Anybody do any PCR in your lab? কেউ কি করেছো?
Yes, sir.
কেউ করো নাই?
স্যার করছি স্যার।
কিভাবে করছে একটা আন্দাজ করো, কি করছিলা?
স্যার একটা টিউবের মধ্যে সব মানে সেট করে তারপর হচ্ছে পিসের মেশিনে দিয়ে দিছি।
যখন করছো তখন কি আইসের উপর রেখে করছো?
স্যার লাস্টের যে মিক্সিং পার্টটা আইসের উপর রেখে করছি। হ্যাঁ
গুড। এই যে জিনিসটা, if you put your samples or your tubes on ice at that very low temperature, ice means 0, you can expect the temperature between 0 to 5 or 7 এর মধ্যে থাকবে। At that very low temperature, you can stop the DNA polymerase to function. That is the thing, very simple. If you do your PCR reaction on ice, you can definitely stop the function of the DNA polymerase. At that very low temperature, like 0 to 4 or 5 degrees centigrade on ice, the DNA polymerase will not make any polymerization, will not do any polymerization. Though this type of hybridization will happen, primer, single nucleotide or double nucleotide, whatever nucleotide they found complementary sequences, they will anneal at that low temperature. But the polymerase, or as the polymerase is inactive due to the low temperature, we can expect primer dimer formation will be prevented. বোঝা গেল? Get it? It's very simple. Reduce the temperature of the room or of the tubes by any way. One way is to put all the tubes on ice. Iceর উপরে রেখে কাজ করো. You can expect primer dimer formation will not happen. Okay?
জি স্যার।
Because at that low temperature, DNA polymerase do not work. At 25, at 30, at 37, they can work very well. Very well means at least like 50% efficiency is good enough to make this primer dimer. Okay, the simple thing you can do in this our type of lab, like we don't have the luxury of buying DNA polymerase, which will not be active during PCR preparation. There are polymerase. I will discuss this later. There are polymerase which is not active during PCR preparation or during normal room temperature. You have to make them active by heating. So when you put the reaction on the machine at 94 degree, they start to active. So at 94 degree, PCR will start, no primer dimer formation will happen. So that is the other thing you can have by having those type of DNA polymerase. Those are called hot start DNA polymerase, those are very expensive. But if you don't have that, you can think of doing all of your work to prepare the PCR reactions at not room temperature, at low temperature, that is on ice. Okay? Any question here?
না স্যার
Okay, now question to you. Do you expect non-specific annealing to the template during this time? প্রশ্ন বুঝছো? Like you are adding your template, you are adding your primer, and do you expect during this PCR preparation your primer bind to the template DNA specifically and also non-specifically? Is it possible?
স্যার স্পেসিফিক্যালি তো বাইন্ড করার কথা স্যার
কেন এটা ভাবতেছো?
কারণ স্যার ওই যে আমার প্রাইমারের সাথে স্যার আমি প্রাইমার তো ডিজাইন করে দিয়েছি স্যার যাতে এমন ভাবে যাতে ও স্পেসিফিক্যালি বাইন্ড করে
Okay, thinking is okay. For me, it is okay as you are beginner. Now think when you are adding the template, most of the cases, what is the status of the template? Double standard or single standard?
স্যার ফার্স্ট স্যার ডিনেচারের সময় স্যার সিঙ্গেল স্ট্যান্ডার্ড হয়ে
No. I am saying about during PCR preparation. When you
Oh,
put
sorry.
your tube on the PCR machine, everything starts. আমি সেই সময়ের কথা বলছি না. I'm not talking about that time. As I said, for like 20 PCR reaction preparation, you may take like 30 minutes. During this time, everything is in the tube. For the first tube, second tube, third tube, whatever. At that time, do you expect your primer to bind specifically or non-specifically to the template? There is no chance because most of the cases, the template you are using is double stranded. As they are double stranded, primer don't have
Primer
any sequence to bind. Okay, but if you prepare your samples like with Chilex method, we discussed before. Chilex method, you have to boil your samples at 100 degrees centigrade, so you make all your DNA free or single stranded. That is different issue. But most of the cases, people are using for PCR double stranded DNA, so you are not expecting your primer to bind to your template specifically or non-specifically. So when then non-specific amplification happen? কখন হয় তাহলে? Definitely during PCR reaction going on. ঠিক আছে? যখন তুমি denature করলা and primers are there to anneal, they can anneal to their specific complementary sequence, they can also anneal to the non-specific sequence. And from both, if you have amplification, you are going to have specific size product and non-specific size product. Okay?
জি স্যার
But during that time, you have already started with the PCR reaction in the PCR machine, you don't expect primer dimer to form because primer dimer are very they can anneal with like only 2 or 3 or only a single base at that very high temperature, 50 degree. What is the lowest temperature you can think of during the PCR? 94, 72, 50. So the lowest temperature is 50. At that 50 is still very high. At that temperature, you don't expect your primer to hybridize with just 1 or 2 nucleotides. So you don't expect primer dimer formation during PCR is going on. You expect this during your PCR preparation. Preparation is সময় এটা হইতে পারে. And to stop that, you just have to prepare all your PCR preparations on ice. That is the cheapest way you can avoid primer dimer. But next class, we will discuss about hot start PCR. Wow, how people are using specific expensive DNA polymerase to avoid primer dimer formation. For today, we are finishing up to here. Okay? Any question?
স্যার এটাই তো স্যার আপনি যেটা বললেন যে স্যার প্রাইমার টাইমারটা হচ্ছে মানে রুম টেম্পারেচারে শুধু হইতে পারে ডিউরিং দ্য পিসিআর প্রিপারেশন বাট পিসিআর রিঅ্যাকশন যখন রান করব তখন লোয়েস্ট টেম্পারেচার 55 পর্যন্ত হইতে পারে এর নিচে তো হবেই না সো প্রাইমার টাইমার হওয়ার পসিবিলিটিটা অনেক কম
একদমই নাই বলা যায়, সাধারণত ওই সময় হয় না। হোয়াট এভার প্রাইমার ডার্মার ইজ ফর্মিং ডিউরিং দ্য পিসিআর প্রিপারেশন, যখন তুমি কাজটা করতেছো, and you can avoid it by just keeping your samples or tubes or ice. And you can, if you can afford, you can buy a hot start PCR polymerase, DNA polymerase. That is different thing.
Okay,
That is different thing. Any other question? If not, then we can stop here today.